Let's talk about Sanger sequencing, also known as the dideoxy chain termination method. This method was developed by scientist Frederick Sanger in 1977, for which he won a Nobel Prize for. This method lets him sequencing the first DNA genome of the bacteriophage phi X. The basic components required are amplified DNA template, DNA primers, DNA polymerase, deoxynucleoside triphosphates called DNTPs, and modified DNTP called dideoxynucleoside triphosphate DDNTP. DDNTPs do not have the 3' hydroxyl group that allows phosphodiester bond to form with the 5' phosphate on another nucleotide to form a chain. This results in termination of the elongation of DNA template at the site of DDNTP insertion. The first step is to heat the duplex to unwind. Then the primers are added to bind to the template strands. Finally, DNA polymerase, DDNTPs, and DNTPs are added and the solution is heated for replication. Now you may ask, how do we visualize these fragments? Sanger initially used 4 separate tubes, each with 4 radio-labeled DDNTPs, and were then loaded in gel electrophoresis. The DDNTPs were read from bottom to top as the smallest fragments closest to the primers traveled the furthest on the gel. Currently, fragments are loaded in a capillary gel and an electric field is applied to separate the fragments by length. Spectrophotometric analysis is used where a laser detects the respective fluorescence of the DDNTPs. A fluorescence is displayed in an electropherogram, and the fragment length allows the order of the nucleotide bases to be distinguished. Hooray! We can now identify daughter strands. However, there are limitations in analyzing long fragments, as sequencing produces less than 1 kilobase of DNA in length. There are also limits in identifying the same nucleotide in a row on a DNA sequence. But emergence of technological advancements provides solutions. Next-generation sequencing, NGS, was the first to sequence efficiently in speed and cost, and development of techniques like PCR improving cost and ability to sequence longer strands. Sanger sequencing has high accuracy rate and revolutionary discovery that is continually used.
Created using Powtoon.